Welcome to this other session of ours. Now we are continuing with our autotrophic nutrition. Now, today I looked at these terms up there, but let me repeat them so they can ask what is meant by chemosynthesis. Okay, sorry, chemosmosis. So chemosmosis, it is the movement of ions across a selectively permeable membrane down an electrochemical gradient. Then somebody can ask you again about the Z scheme, the one we have looked at up there. So we said that it is a diagrammatic representation of electron flow in cyclic phosphorylation and non-cyclic phosphorylation, showing the changes in energy potential of different electrons. I will not waste time on this one because we looked at it up there when you are looking at the Z scheme. Now we are going to look at what we call the comparison of cyclic and non-cyclic phosphorylation or photophosphorylation. Now when they talk about comparison, this question mostly come in section B number 2, even number 1 paper 2, even number 1 in paper 2 graph work. When they say compare, comparison means you are going to give both the differences and the similarities. But when you are giving the similarities, what you should put in your head is that the any point you write there begins with what? With the both. For example, our first comparison here or similarity, we are saying in both there is flow of electrons through several electron carriers, as we have seen above there. Our second point is that in both there are pigment systems which accept and lose electrons. So when we talk about pigment systems, we looked at them. Those are the photosystems 1 and the photosystem 2. Then our third one similarity is that in both ATP is formed. Then our next point, we can say that in both the pigment system 1 is involved. The pigment system 1 involves 5 electron movement, is located in the thylakoid membrane. Then our number 6 point, we are saying in both protons are moved outwards of the thylakoid. Say in both protons, in bracket the hydrogen ions, are actively pumped from the stroma into the thylakoid space. I talked about this. The number 8, we are saying that in both there is photo excitation of electrons in the pigment system. Now that has been our similarities, and we have given you almost 80. We have given you almost 8 similarities. Now we can go to differences. When you are answering differences, sorry for that, but when you are answering differences, we use the word while. So when you are answering paper 1, when they say give the differences, you say that's where you use a table to simplify. But when it comes to paper 2, please don't draw a table. But use the word while. Now we have non-cyclic phosphorylation and cyclic phosphorylation. Then we are saying our first bullet is that the electron flow unidirectionally, in bracket the non-cyclic. Then here electrons flow cyclically, as we have seen above. Unidirectionally means in one direction. Then our second point is that first electron donor is the source of electron, which is water. We saw there in photolysis. Then this side we are saying that the first electron donor is the pigment system 1. Remember we began with the P700. Now this side the last electron acceptor is the NADP. Then this side the last electron acceptor is the pigment system 1. So we are saying that this side, the non-cyclic, we are saying that the products are ATP, NADPH, and oxygen. Well, the other side we say that the product in cyclic is only ATP. So this side, differences of non-cyclic, we are saying that it involves both pigment system 1 and 2, while the other side, for it involves only the pigment system 1. Now this side, we are saying that photolysis of water occurs. Then in cyclic, we are saying no photolysis of water. I also talked about what? Photolysis. So let us go and look at what we are calling the role of water in photosynthesis. Now we are saying that the catalytic photolysis, in bracket, splitting or breaking of water, produces electrons and protons. Now water is a source of electron to replace those lost by chlorophyll or that lost by the photosystem too. Then water is also a source of hydrogen ions needed to produce the NADPH. Then another one, water is a source of hydrogen ion, which when flowing from the thylakoid space into the membrane via the ATPase, it forms what we call ATP. Then we are saying that water is a substrate, reactant, raw material for photosynthesis. Then lastly, water is transparent, so photosynthesis can take place underwater. Stroke light can penetrate to what? To chlorophyll. So allow me end here. So we meet in our next session. So that has been the light dependent stage or the light phase. So we are going to meet in the darker reaction, or what we are calling the biosynthetic phase, or the Blackman's reaction. I sign out.